Hello, I'm Anne Montgomery, Editor-in-Chief of Bioprocess International magazine. I'm here at Biotech Week in Boston speaking with several thought leaders about advancements in bioprocessing and cell therapy development. I hope you enjoy their insights and their visions for the future.
Hi, so my name's Christopher Bravery. I'm from Consulting on Advanced Biologicals. So I'm a consultant. I'm an ex-regulator in Europe. Used to work in broadly across all biologics. So scientifically, I've always found cell therapies, what we call advanced therapies in Europe, much more interesting than traditional protein therapeutics, but I've worked a lot on those. So stability is a topic I've raised this year because when I look at stability studies for my clients, they don't really fit with the recommendations that are in the harmonized guidance of ICH. So if you compare to protein therapeutics, we obviously have a much deeper understanding of the potential implications of stability. The potential outcomes. So with protein therapeutics, you can get aggregation of the proteins during the shelf life. You can also get interactions with the excipients that lead to aggregation or denaturation, oxidation, etc. And those have known clinical impacts. So they can lead to immunogenicity, they can lead to loss of efficacy, etc. With cell therapy, our understanding of this science is still very early. And what we don't really know is what the potential implications are of loss of stability. And then added to this, they're very complicated, much more complicated than proteins. So at the moment, what I see predominantly is the heavy reliance on viability. The assumption being that the cell needs to be alive to have its action, which in most cases will be true. But the sort of the classic methodology that's used is trypan blue. And everybody knows trypan blue is not particularly good. And the problem is it really measures the terminal event of the cell having lost its viability. So I've been actively trying to encourage people to use other markers of viability. So there's lots of markers that will pick up early events that lead to loss of viability. Things like annexin staining, for things like apoptosis and so on. And in fact, there's something ridiculous like 16 ways a cell can die. And yet this isn't really explored very much within the community, although many people are aware of this. So I think that's one element. But really the biggest problem I see, I think, is that unlike in the protein therapeutic world, where it's well understood that the first thing you do before you develop a stability specification is you degrade your product. And you characterize the degraded product to understand what will happen. So we call this stress testing. And you can do that by elevated temperature, bubbling oxygen through it to oxidize it, photostability, you shine bright light on it, and so on. There are a whole range of different methods. I'm not seeing this done currently in the cell therapy community, so my usual criticism of a stability specification is where's the evidence that the methods you're using are stability indicating? So I think this is a gap. This is a gap in the analytics that we need to think about what other methods might be stability indicating. We need to explore that aspect of our products. My ideas and thoughts in this area beyond other methods of looking at cell viability are things like more biochemical measures of cell health and activity, if you like, and so on. You do always include your potency assay in stability studies. Potency is also a horribly difficult topic for cell therapy. We're not looking at a product that has a single mechanism of action. It has multiple mechanisms of action. It's dynamic. So developing potency assays is also difficult. And they're not always particularly sensitive methods. And for stability, you want a sensitive method because you want to be able to see changes in stability early. So on some occasions, people have found their potency assays are stability indicating in that they see a change in the potency assay before, say, viability changes. But they may not be the ideal method. But then it becomes more difficult because unlike proteins, cells are resistant to oxidation because they're alive. So there is a bit of a gap there, and it's possibly something that's needed for basic science to go and look at cells and think about ways that would be useful to measure stability in cells, and perhaps develop some new and more useful methods. But right now, I think the whole industry is a bit stuck with what's the right way to do this and what are we missing? So, I think the take-home message is that we need to embrace this concept of forced degradation, of stress testing, to understand what happens to the cells under stress, and look beyond viability and look at other measures, be they biochemical, molecular, or whatever. Cells have receptors. Many of those receptors may be involved in your mechanism of action. If those receptors are upregulated or downregulated, then that could be an indicator that the cell's losing its quality, basically. There's another classic problem, actually, with looking at viability, and that's to forget to look at the overall cell content. It's really quite surprising the number of times I see a specification that shows viability must be above 70% or something like that, but there's no total measure of cell number. So you could apparently have a level viability, but actually your cell number could be declining. But in general, there are a myriad of other issues around cell therapy. So you have two basic choices you need to make early in development, which is, will I provide this as a fresh product that is at room temperature or refrigerated temperature and ship it to the patient? Now, we know that it's very hard to get a shelf life beyond 48 hours, maybe a little bit longer than that in a few occasions. So then the other choice is to cryopreserve. And at the moment, our options for cryopreservation kind of pretty much hang around using DMSO to protect the cells from damage. And rather little has been done on other sorts of excipients that could protect against damage and freezing. And while DMSO isn't actually particularly toxic, it can be, depending on the level, occasionally it can lead to hypersensitivity reactions. And it's a general principle with regulation that you try not to give more of anything than you need to. So the challenge of trying to keep the DMSO level low, either by reducing it in the formulation or maybe even diluting the formulation, so freezing the product more concentrated and diluting it and infusing it or whatever. So when you're looking at stability at liquid nitrogen temperatures, you kind of don't really expect much to happen. So, however, I think clinically it's known that for transplantation of bone marrow that it isn't an infinite amount of time the cells can exist in the freezer. You also have to consider the container and the closure system and whether that's capable of lasting for more than a few years in the freezer without being breached, because you might lose sterility. So there are a myriad of issues around how to present your product, whether it's fresh, frozen, and there's pros and cons to both. I mostly see cons to both, actually, neither is ideal. So it would be great if one day someone had developed a way of sort of, I'm trying to think of the phrase, I've lost it. It would be great if one day people could develop a formulation that would allow you to keep the cells in a liquid formulation for maybe more than a week or something like that. In some cases, if you're looking at allogeneic products where you want to make a large batch, then you may still cryopreserve, but if you could thaw sub-batches and then have them in a form that was stable for a few weeks, this would make a huge difference to the commercialization of the product. So I think there are a lot of issues around how we formulate. And at the moment, most cell therapy developers really are just looking for off-the-shelf solutions to cryopreservation or formulating their product. They're borrowing ideas from each other. And I see rather little formulation development. And so when you reflect back on protein therapeutics, quite a large amount of work is done for every new protein therapeutic on its formulation, because each protein is slightly different in the same way as each cell will be slightly different. And you need to consider the sort of optimum formulation to get the shelf life you want in the form that you want. And these sort of studies aren't done yet really systematically with cell therapy. It's a little bit more of a sort of select a couple of formulas and hope that one of them looks good, and if it looks good, let's move forward. So another gap, I think, for the basic science to try and work more on how to preserve cell therapy products in a form that's clinically suitable, that can be administered to the patient safely, and provide longer shelf lives as well. I presented an example of a typical specification for stability that I see, an invented example. And it contained an identity test, which actually isn't even required. Appearance, well, of course, you always include appearance, the potency method and viability, and kind of that's it. And it may turn out that's sufficient, but right now we don't really know. And when I compare it to a protein therapeutic, it seems thin, rather lacking in detail. And I think, part of the problem is it's overwhelming how many things you could look at with a cell. There's all the surface proteins, there's all the intracellular proteins, there's the DNA inside, whether you've got molecular stability, which could be an issue with freezing, and so on. So it's almost overwhelming how much we could look at, and the question is, well, what's relevant? And we don't know yet.